Welcome student, I am Dr. Rashmi Verma, working as assistant professor in the School of Biotechnology, IFTM University, Muradabad. Today we are here to discuss an important technique that is Agarose Gel Electrophoresis. Agarose Gel Electrophoresis is basically a tool to separate DNA or RNA based on their fragment size. It is used in the field of biochemistry, molecular biology, genetics, and clinical chemistry. What is agarose? Agarose is isolated from seaweed, Gracilaria, and Gelidium. It consists of repeated agrobio subunit, that is D and L form of galactose. During the process of gelation, agarose polymer associate non-covalently and form a network of bundles. The pore size of the gel determine the sieving molecular properties of the agarose gel. What is the principle of Agarose gel electrophoresis? It separates DNA fragment by size in a solid support medium such as agarose gel. The sample, that is DNA, are pipetted into the sample wells followed by application of electric charge, which causes negatively charged DNA to migrate towards the positively charged anode. The rate of migration is proportional to the size of the pore present in the gel. The smaller fragment move more quickly and wind up at the bottom of the gel first. The DNA is then visualized by including gel and intercalating dye, that is ethidium bromide. The illumination with ultraviolet light causes intercalated dye to fluoresce. Instrumentation of Agarose gel electrophoresis. It consists of electrophoresis chamber and a power supply. Gel casting tray that is made up of ultraviolet transparent plastic. Both end of the tray is closed by a tape when the gel is being casted and removed after the gel solidifies. Sample combs. These combs are placed in the molten gel and removed when the gel solidifies. This comb is used to make the wells in the gel. Electrophoresis buffer. Two types of electrophoresis buffers are used, TAE buffer and TBE buffer. TAE stands for tris acetate EDTA buffer and TBE is tris borate EDTA buffer. Loading buffer. It contains something dense, that is glycerol, to allow the sample to fall into the sample wells, and one or two tracking dyes that allows the proper monitoring of the DNA into the gel. Staining. Staining is done by ethidium bromide dye that intercalates in the DNA molecule. Transilluminator, or it is also called ultraviolet light box, that is used to visualize the stained DNA in the gels. This slide shows the basic steps involved in the electrogel electrophoresis. Here you can see first step is DNA sample loading, second is application of electric field, third is DNA separation and UV transillumination and documentation. First we talk about DNA sample loading. DNA is mixed with the tracking dye and loading buffer, and this is then placed in the wells made in the gel by gel comb. Okay, now you can see DNA is negatively charged. It travels towards the positively charged anode when electric field is applied, and the DNA fragments get separated on the basis of their size, and a marker DNA is also loaded here so as to determine the size of the fragment. Now this gel is placed in gel dock or UV transient illuminator to check the bands. Steps involved in agarose gel electrophoresis. To prepare a gel, agarose powder is mixed with electrophoresis buffer to the desired concentration and then heated in a microwave oven to melt it. The concentration of agarose gel depend on the size of fragment to be resolved. The concentration of agarose is referred to as percentage of agarose to the volume of buffer, that is weight by volume. And agarose gel are normally in the range of 0.2 to 3%. Lower the concentration of agarose and faster the DNA migrate because the pore size increases, so to facilitate the movement of DNA fragment. After melting the DNA in a microwave, ethidium bromide is added to the gel so as to make the final concentration of 0.5 microgram per ml. That will facilitate the DNA visualization after electrophoresis. After cooling the solution to about 60 degrees Celsius, it is poured in the casting tray containing the sample comb and allowed to solidify at room temperature. After the gel has been solidified, comb is removed and taking care not to rip the bottom of the wells. Gel is then placed in a plastic tray and is inserted horizontally into the electrophoresis chamber and is then covered with a buffer. After addition of the electrophoresis buffer in the electrophoresis chamber, sample containing DNA mixed with loading buffer are then prepared into the sample well. The lid and the power leads are applied to the apparatus and the current is applied. The current flow can be confirmed by observing bubbles coming off from the electrodes. The DNA will migrate towards a positive electrode, that is anode, marked red. The distance DNA has migrated in the gel can be judged visually by monitoring the migration of tracking dye, that is bromophenol blue and xylene cyanol dye. Application of Agarose Gel Electrophoresis. First application is DNA RNA analysis, such as PCR, DNA sequencing, and RNA isolation. The techniques can be used to separate DNA fragments based on their size, which is essential for downstream applications such as cloning and gene expression analysis. Second application is protein analysis. It involves the separation and analysis of proteins. The separation of protein based on their size and charge can provide insights into the structure and function of the protein. Third application is forensic analysis. This is used to identify DNA sample from the crime scene. The technique is used to separate DNA from different individuals and then compare them to the DNA sample collected from the crime scene. Fourth application of the Agarose Gel electrophoresis is in medical diagnostics. It is used to identify genetic mutation associated with various diseases such as sickle cell anemia and cystic fibrosis. Advantages of Agarose Gel electrophoresis. For most of the application, only a single component, that is agarose, is needed. No polymerization catalyst is required for the polymerization of gel. So we can say agarose gels are simple and rapid to prepare. The gel pour does not denature the samples and the samples can be easily be recovered. At last, we will discuss about the disadvantages of agarose gel electrophoresis. The gel can melt during the process of electrophoresis while running the gel, and buffer can also become exhausted, and different forms of genetic material may run in an unpredictable forms. So these are the disadvantages of agarose gel electrophoresis. Thank you.